Hi there, my name is Paul Jaschke. I'm a lecturer in synthetic biology at Macquarie University. And I'm going to tell you stories, a couple of different stories today from the lab where we've been refactoring phage. So the main host of the phage that we work on is E. coli, which is only a fairly minor component of the healthy gut, but under inflammatory conditions, this can increase and you can have blooms of E. coli in the gut. And these blooms can also then make you susceptible to other pathogens such as Clostridium difficile and Campylobacter. And these blooms can occur with even simple antibiotic treatment. So the bacteriophage that I'll tell you first about is called Phage 174, which infects E. coli of the human gut. And it makes an icosahedral capsid shown in the upper right here. And if you drop that onto a plate of bacteria, it will make a clearing called a plaque where all the cells have been killed. And that process is shown in the video in the middle here, sped up quite a bit. So Phage is interesting in that it's a member of the microviridae, which are a dominant and persistent member of the human gut virome. And as well, Phage is particularly interesting to us because it seems to exclusively infect E. coli strains associated with the human gut. And this is done via the LPS or lipopolysaccharide receptor, which is quite diverse across different strains of E. coli. And we have another project that I won't talk about today, looking at modifying that host range. One of the interesting things that makes it also a little bit difficult to use Phage is that its genome contains a lot of overlaps. So shown here, gene B is overlapped with A and A star, and gene E is overlapped with gene D. So to be able to make Phage sort of a chassis from which to address some of these commensal and pathogenic E. coli strains that we're interested in targeting, one of the first projects I did many years back now is to remove all these overlaps so that we could manipulate each gene independently. And that's shown in this process here. Gene overlaps have a lot of different functions across bacteria and eukaryotes and viruses. And some of those could include unwinding the RNA structure such that the downstream gene has less structure around its ribosome binding site and start codon. As well, it can increase the concentration of ribosomes around the downstream gene start codon. It can protect genes from mutation, and as well, it can just merely encode more information by having in a finite amount of nucleotide space. And this is sort of shown in the Phage genome with gene E, which is embedded entirely inside another gene, and gene E is the lysis gene, and it's a younger gene, so it had developed later in evolutionary history. So once we created this decompressed phage, we found that it had lower fitness compared to wild type, and that was shown by reduced burst size, so less phage popping out of every infected bacteria, and consequently a lower plaque size. So we tried to understand why that was. What did we mess up by removing these gene overlaps? And one of the first hypotheses we had was, did we change the shape of the capsid? And so we looked at that using electron microscopy and showed that it essentially is indistinguishable from wild type. But one thing we did find was that the assembly pathway of these virions is disrupted by a lowered expression of gene B. And so you can see gene B in the wild type is embedded here in A and A star, but in our refactored or decompressed phage, it's downstream of gene A, and now it's been separated from its promoter. Likewise with gene C, which is a protein that's quite important in switching from single-strand DNA synthesis to double-strand DNA synthesis, and then packaging of the single strand into the virion. Its location changed from downstream of A to being now 2 genes downstream of A. And its expression was also decreased with proteomics. So what we learned from this is that 5' untranslated regions and transcript boundaries probably do have some dramatic effects, apart from just the disruption of the overlaps. And as well, protein measurements may be necessary during the tuning process. Several outstanding questions we have are, sure, the virion looks the same, but does it function dynamically the same? Does it eject the genome in the same way? And we're looking into that, as well as, can we just restore fitness by rationally tuning these ribosome binding sites that are now separated from the other genes? Can we simply tune those to increase fitness back up to wild type levels? Second story I want to tell you about with refactoring is a project I did to establish the absence of function. So in the top shown here is our sort of naive idea of how we can design a construct by putting 3 different genes side by side, and they all express only their own protein. But in reality, a lot of genes contain hidden or cryptic genetic information to produce a protein or an RNA, or sometimes to encode structure of the RNA and things like that. So can we do a better job of our refactored genomes looking like our designs? And so I looked again at the Phage genome, this time back at the wild type, and looked at cryptic open reading frames that were conserved across the other microviridae, and found nearly 100 of them. And so I set about erasing these by essentially disrupting them using single point mutations that didn't change any of the overlapping Phage gene proteins. And after erasing nearly all of them, we found that only one of the changes we made seemed to have any phenotypic effect. So by this way, we sort of demonstrated by synthesis that genome annotation was essentially complete for most of these open reading frames. And lastly, a project that's still just starting in the lab is to see if we can make phage safer to use as phage therapy vectors. So one of the issues with using engineered phage is just that uncontrolled release factor of it. Once you give it to a patient, it's sort of released into the environment. So could we make it a little bit more contained using essentially an engineered tRNA here, shown called the amber suppressor initiator tRNA, which we've modified the anti-codon of this tRNA to recognize, instead of the regular start codon ATG, it recognizes the TAG, which is normally a stop codon in E. coli. And so we express this in an engineered phage, in the same cell that we infect with an engineered phage, where the start codon of gene F, which is the major capsid protein and required for virion assembly, has been changed to TAG stop codon. And thus, in the cell that's expressing that tRNA, we can amplify that phage quite easily. But then when we were to take it and say use it in a therapeutic context where the natural bacteria do not contain this engineered tRNA, the phage will still inject its genome as usual and will express all of its genes except for the gene that has that amber stop codon put in place of its start codon. And so it will not make one of the important products, and thus it will still express its lysis gene and lyse the bacteria, but it will not spread any further. And so in this case, it doesn't get that benefit of amplifying like a regular phage does, but it does have that act more like a smart antibiotic. This is just to show that we've been able to do that. Pamela Sumbris and Russell Vincent did that work during their theses. So I'd just like to say thank you and wrap it up here. Thanks to Adrian and John and Erica for doing that decompressed phage work during my postdoc at Stanford, and as well Diane, Kay, and Gabrielle Dotson, who helped with that PNAS paper that I described. And here's Pamela, who's going to start PhD in a few months, hopefully in the lab, and Russell, who is just finishing up his PhD. Bradley Wright did all the proteomics work and spearheaded that, looking at the decompressed phage genome, along with Dr. Ruan and Professor Mark Malloy, who were instrumental in helping with that, as well as the assistance of APAF to do all those experiments. All right, thanks a lot, and I'll take any questions.